In Trenton, New Jersey, a newlywed woman is told she is infertile. She and her husband had planned on a large family. In Edinburgh, Scotland, a middle-aged couple remarried decided to have children, but are found to be incapable of conception. In Vancouver, Canada, a couple who had tried in vitro fertilization twice returns to their clinic for the third and final try. Each of these couples has decided upon the most monumental decision of their lives, the extension of their love for one another into the life of a newborn child. Unfortunately, their doctors have diagnosed each woman with a condition called infertility due to anovulation. Simply put, these women do not properly produce eggs, and therefore pregnancy is unlikely. There are several causes for this condition. In many, it starts in the pituitary gland at the base of the brain, where hormones called gonadotropins are produced. These include follicle-stimulating hormone, or FSH, and luteinizing hormone, or LH. At birth, a female's ovaries contain about 500,000 immature eggs, of which only a fraction mature during a woman's lifetime. Each egg is surrounded by a group of smaller cells and is called a follicle. FSH causes the follicles to grow. An egg also begins to mature at this time. At about day 14 of the average adult female cycle, the pituitary gland produces luteinizing hormone, or LH, which stimulates the release of the egg from the follicle and the ovary. Both FSH and LH must be present in the right amounts at the right time to cause the release of an egg, and if sperm is present, an eventual pregnancy. If FSH is not normally present in the woman's ovaries at the right time and amount, the egg is not produced and pregnancy cannot occur. This condition, anovulation, causes infertility. In the late 1920s, biologists discovered that by injecting gonadotropins at just the right time, the follicles of the egg could be artificially stimulated. This discovery led to the hope that fertility could be enhanced by the timely administration of gonadotropins. But gonadotropins are difficult to obtain. To satisfy the needs of these couples and millions of others around the world, scientists have labored for most of this century to find a pure, reliable, plentiful, and safe source. This is their story. Prior to the discovery of gonadotropins, infertile couples suffered derision, condemnation, or pity. Often the cause of infertility was blamed on the woman. The consequences were often severe. Later, science replaced superstition, and infertility, defined as an inability to conceive a child after 12 months of unprotected sexual intercourse, was found to occur in about 15% of all couples desiring children. Of these, roughly 40% are due to female infertility alone, 40% to male infertility, and 20% due to complications of male and female infertility issues. Research in the process of artificially causing ovulation with gonadotropins began in the 1940s. The process required often painful intramuscular injections, beginning on the first day of the woman's cycle and continuing daily until day 14, when an additional injection of a hormone called human chorionic gonadotropin, or HCG, was injected. While an inexact method, somewhat satisfactory results were obtained, and the demand for gonadotropins grew. But gonadotropins are difficult substances to isolate in nature. In the 1950s, just a few grams of gonadotropins required hundreds of meticulous extractions from upwards of 27,000 liters of raw material. And the raw material, the only source of this wonder drug, was repugnant to most people: the urine of pregnant horses. From the 1920s until the 50s, millions of gallons of horse urine were collected through a vast network of farmers who regularly stalled their mares while pregnant in special paddocks equipped with collection troughs. From these troughs, farm hands would convey the urine into special cans, which were periodically collected by pharmaceutical field agents. At centralized collection locations, the agents would decant their collections into large drums for shipment to pharmaceutical processing plants. Here, the urine would be meticulously filtered and the active gonadotropin protein extracted through a series of exhaustive distillations. And yet, after all the effort, the resultant injections of gonadotropins were not always successful. Variations in purity, specific activity, commonly referred to as potency, inconsistent batches, and seasonal variations in supply drove researchers to seek improved sources of gonadotropins. In 1947, Dr. Piero Danini, an Italian scientist, was able to extract and purify gonadotropins from a new source, human donors. Similar to the process used in harvesting the hormone from animal donors, the urine of postmenopausal women introduced commercially in 1960 provided a higher quality source of gonadotropins. But the resulting product was not successful in causing a birth until 1962. As the demand for gonadotropins grew, pharmaceutical companies searched relentlessly for some means of creating gonadotropins from a chemical process.
Research and development for a new drug is a process of 12 to 15 years in collaboration with universities, academic hospitals, and scientists from all over the world. Compounds under development are constantly assessed for efficacy, acceptability, and safety. The testing process is rigorous. From over 5,000 compounds, only one or two might finally reach the doctor and the patient.
Then, on April 2nd, 1953, scientists were given a major breakthrough that would revolutionize biological science for all time. Two genetic researchers, James Watson and Francis Crick, elucidated the structure of deoxyribose nucleic acid, or DNA. DNA, found in the nucleus of every living cell, was discovered to be both the blueprint and the operating program of every cell in the body. DNA determines what a cell will become and what it will produce throughout its life cycle. Scientists excited by this discovery theorized that one day a strand of DNA could be created that would drive a simple cellular organism isolated in a laboratory to do nothing but produce large quantities of gonadotropins. Even more exciting was the prospect that this process could produce specific gonadotropins such as FSH, which was thought to be more effective in achieving pregnancy than generic gonadotropins. Such a development could literally place nature's factory in the hands of humankind, enabling scientists to treat a wide range of medical conditions. After nearly 20 years of research, in 1972, scientists finally managed to synthesize some simple hormones and proteins such as insulin in quantities which were practical for mass production. The new process was called recombinant DNA technology. But even as late as the 1990s, it was considered highly improbable that FSH could ever be synthesized. Dr. Weber Oleva.
The pregnancy hormones are the most complex hormones known to date. And they're so very complex that they cannot be synthesized by organic synthesis.
But Dr. Oleva was wrong. Ironically, just a few years after this interview was filmed, his own research team at Organon, a Dutch pharmaceutical firm, commercially perfected the process for synthesizing FSH. Using recombinant DNA technology, researchers employ a common bacterium or virus called a vector. In nature, such an organism invades a cell and attaches its DNA to the host cell's DNA. Thereafter, the host cell follows the instructions of the invader's DNA, becoming the slave of the invader. Normally, we associate viruses and bacteria with harmful activities, but the same vector can be used for beneficial purposes. Today, scientists are capable of stripping out the DNA of viruses and bacteria and substituting their own DNA. DNA that drives the host cell to do nothing more than produce FSH. Before introducing the vector containing the foreign DNA into host cells to produce FSH, it must be cloned. Since the clone is precisely identical to the original DNA, the resultant FSH is the purest form of the hormone in existence. Once the vector is isolated in large quantities, it can be introduced into the desired host cells, such as mammalian, yeast, or special bacterial cells. The host cells will then synthesize pure FSH from the implanted DNA. Because genetically produced FSH is chemically identical to the original sample, it is purer and more efficient than FSH derived from raw animal or human sources. Genetically produced FSH can be harvested in a laboratory setting such as this one in the Netherlands. This recombinant FSH can be produced in large amounts, sufficient to easily satisfy the needs of infertile women throughout the world. Genetic engineering produces FSH that offers significant advantages over FSH extracted from urine. Its high purity and high specific activity or potency allow FSH, which is obtained through recombinant DNA technology, to be self-administered at home using smaller needles and shallower, less painful injections. Because recombinant FSH is synthesized in genetically modified laboratory cells, each preparation of the product is identical to the last, ensuring batch-to-batch consistency. Finally, there is no dependence on urine collection. A steady supply of recombinant FSH is always assured. Today, the contribution of genetically engineered FSH has demonstrated significant success rates. These success rates include both in vivo fertilization, that is within the woman's body, and in vitro, or assisted reproductive technology, ART. ART involves removal of the mature egg from the woman, fertilization in a clinic, and then return of the fertilized egg or embryo to the womb for gestation. Drugs containing FSH have been successful in treating infertility in women who do not ovulate and in women who are enrolled in an ART program. In ovulation induction therapies, recombinant FSH has led to significant success rates for infertile couples. Ultimately, employing the growing knowledge of the human genome, scientists will discover the underlying genetic causes of infertility and correct them in both the affected population and those of their children for generations to come.